All right, welcome to the Explainer. Today we're diving into a real paradox of the microbial world, an organism with a split personality, Clostridium sporogenes. It's this fascinating bug that can be both a friend in our gut and a pretty dangerous foe. And this quote really just nails it, doesn't it? A potentially lethal pathogen and a beneficial commensal. So in one situation, it's helping out your immune system. In another, it's a serious, life-threatening problem. For anyone working in a clinical microlab or in infectious diseases, getting a handle on this duality isn't just interesting, it's absolutely crucial. So before we really get into the weeds here, just a quick heads up on how we put this all together. This whole explainer was built using a Gemini deep search to pull in the latest data. All the amazing images you'll see are from our friends at Microbiology. And hey, if you're a data nerd like me and want to check the primary sources, we've got a full notebook document available for you to review. Okay, so let's dig into this bacterium of two tails. We're going to start with its identity crisis, and that begins with where it sits on the family tree. Formally, it's in the family Clostridiaceae. And more specifically, it's part of what we call Clostridium sensu stricto, the true Clostridia. And here are those two faces right next to each other. It's like Dr. Jekyll and Mr. Hyde. On one hand, you've got the beneficial commensal. This is the good guy, living in your gut, taking tryptophan and turning it into something called 3-indole propionic acid, or IPA for short, which is a really powerful antioxidant. It even helps tune up your immune system. But then, you've got the other side. The opportunistic pathogen. When things go wrong for the host, this same bug can cause some truly nasty stuff. We're talking bacteremia, myonecrosis, just devastating infection. Now, this is where it gets really tricky, especially for the lab. Think of Clostridium sporogenes as the diagnostic twin of group I Clostridium botulinum. I mean, look at that footer. They share more than 99% of their 16S RNA gene sequence. They're practically identical. The one single, critically important difference, Clostridium sporogenes is missing the genes to make the botulinum neurotoxin, one of the most potent toxins known to humanity. This is a massive public health issue. So that brings us right to the lab bench. How do you deal with this diagnostic dilemma? Because let's be real, getting this wrong, well, it has huge public health consequences. Okay, so when you get this on the bench, it does have some classic tells. You're looking at these large, gram-positive rods. You'll see those oval, subterminal endospores that make the cells swell up. On a blood agar plate, you get these cool, kind of scary medusa head colonies, and most strains are beta-hemolytic. And if you put it in a cooked meat medium tube, whoa, you'll see a ton of gas and proteolysis. The problem? As classic as this all looks, it's just not enough to confidently separate it from its much more dangerous twin. And this is why choosing your ID method is so important. Your standard 16S sequencing, the workhorse of microlabs everywhere, it's totally useless here. That greater than 99% similarity means it just can't tell them apart. No, the undisputed gold standard, the real hero of this story, is MALDI-TOF mass spec. It doesn't look at the genes, it looks at the proteins. It creates a unique protein fingerprint that can tell these two apart with certainty. It's the only way to get a secure species-level ID. All right, so beyond MALDI, the biochemical profile can give you some more clues. It's going to be negative for catalase and oxidize, but it loves to break down fats, so it's lipase positive, and it's also positive for gelatin hydrolysis. Now, here's a key test. It's negative for lecithinase. That's super helpful for telling it apart from another nasty Clostridium, Clostridium perfringens. The indole test, well, that one's a bit tricky. It's listed as variable, and you see some conflicting reports in the literature, which probably just comes down to differences between strains. So that's the what. Now for the how. How does this bug that's just hanging out in a lot of our guts, causing no trouble, suddenly turn into an invasive pathogen? The secret isn't that the bacterium gets stronger. It's that the host gets weaker. And when we ask who is at risk, the answer is pretty clear. These infections are almost always endogenous, meaning they come from the patient's own body, their own gut. It happens when the body's defenses are just shot. And if you look at this list, it tells the whole story. We're talking about patients with cancer, a hole in their gut, or large areas of dead tissue, maybe from a pressure sore.
It's all about a compromised host creating the perfect storm for this bug to invade.
A really good way to think about this is what's called the two-hit model of infection. It's a 1-2 punch. First, you need hit number 1, a barrier breach. Something tears a hole in the gut lining, or you get a really deep wound. This lets the bacteria escape from where it's supposed to be. Then comes hit number 2. It needs an anaerobic niche. It needs a place with no oxygen to grow. That's the perfect little oxygen-free haven for those dormant spores to wake up, germinate, and start multiplying like crazy.
Okay, so when this does happen, how do we treat it? These infections are no joke, so you have to be fast and you have to be decisive. The strategy is really a two-pronged attack. Hit it hard with surgery, then follow up with the right antibiotics.
And let me be crystal clear about this. The first step is absolutely non-negotiable: aggressive surgical source control. You have to get in there, cut out all the dead tissue, and drain any abscesses. Why? Because antibiotics just can't get into those areas. If you don't clean out the source of the infection surgically, the treatment is just going to fail. Period.
Now, what's interesting is that there aren't any specific guidelines just for Clostridium sporogenes. So what do? We treat based on the clinical syndrome. For example, if we look at the Dutch swab guidelines, for a complicated intra-abdominal infection, they might recommend a combo like cefuroxime plus metronidazole. But if you're dealing with something as scary as a necrotizing soft tissue infection, you're going to escalate to broader spectrum drugs, like meropenem. And this lines up pretty well with international guidelines, like from the IDSA. For a high-risk intra-abdominal source, something like piperacillin tazobactam is a solid choice. But again, for necrotizing soft tissue infections, they go for the big guns: a carbapenem plus vancomycin, and, this part is key, clindamycin. And they add clindamycin specifically because it's thought to help shut down toxin production, which can be a huge deal.
What about for kids? Well, the game plan is pretty much the same, but the details are everything. It's all about precision. Every single dose has to be calculated based on the child's weight. You can see some typical weight-based doses here for the go-to drugs. But let's be honest. If you're managing an infection this severe in a child, you are absolutely calling a pediatric ID specialist for a consult. That's a must.
Okay, so what about susceptibility testing? Here's another little wrinkle. UCAS doesn't actually have specific breakpoints for Clostridium sporogenes. So the standard practice is to use the breakpoints for Clostridium perfringens as a surrogate. Now, for the most part, it's usually susceptible to drugs like penicillin and metronidazole, but you can't assume. It is so important to actually test your clinical isolate because resistance can and does pop up. All right, last but not least, prevention. How do we stop this thing from spreading around the hospital? Well, the key word here is spores. Because it's a spore former, we don't have to reinvent the wheel. We just borrow the playbook we all know for fighting that other infamous spore former, Clostridium difficile. And those core measures are totally non-negotiable. First up, you've got your standard contact precautions: single room, gowns, gloves, the whole 9 yards. Then you have to clean the environment, and not just with anything. You need a sporicidal disinfectant. We're talking bleach. And finally, there's antimicrobial stewardship. This one is huge. Using antibiotics wisely helps protect the patient's gut microbiota in the first place, which is our best defense. Which brings me to maybe the single most important and, honestly, most frequently messed up part of controlling spore formers: hand hygiene. You've just left the patient's room. What do you reach for? The alcohol-based hand sanitizer, or do you head to the sink for soap and water? The answer, 100% of the time, is soap and water. I can't say this loudly enough. Alcohol does not kill these spores. It just doesn't work. You need the soap, the water, and the physical friction of scrubbing to literally wash the spores off your hands and down the drain. This isn't just a best practice, it's a fundamental rule of patient safety. So all this leaves us with a really interesting, kind of provocative question for the future, right? We're heading into this new era of microbiome medicine. We know this bug, Clostridium sporogenes, can make beneficial stuff, but can also be a killer. So the big question is, how are we going to figure out how to balance that? How do we harness the good without unleashing the bad?
Okay, that's a wrap. But please remember, this explainer is strictly for informational purposes. If you need any kind of medical advice or a diagnosis, you have to go see a qualified professional. That being said, we'd love to hear from you. What are your experiences with this fascinating bug? Drop your thoughts and feedback in the comments below.